Hi everyone, my name is Aarthi, and today I'm going to be describing some new findings for pancreatic adenocarcinoma that were really uniquely driven by recent advancements for single-cell analysis from our lab. This is a joint effort by the Muzumdar lab at Yale, as well as my PhD lab led by Smitha Krishnaswamy. So in this study, we used a suite of recently developed deep learning tools for single-cell analysis, where from single-cell RNA sequencing data alone, we sought to map cellular heterogeneity within cell types, derive cellular dynamics and regulatory behavior, and compare our findings across diverse settings. Today, I'll be discussing these models, as well as how the predicted cellular and molecular mechanisms were then validated in vivo and in vitro in the context of obesity-associated pancreatic cancer. So the pancreas is comprised of the exocrine and the endocrine compartments, and it's recently been appreciated that crosstalk between these compartments is highly relevant in many pancreatic diseases, including pancreatic ductal adenocarcinoma. Recently, our collaborators in the Muzumdar lab showed that in a mouse model of obesity-associated pancreatic cancer, obesity plays a causal role in early PDAC progression, driven by the aberrant expression of peptide hormone cholecystokinin, where in pancreatic beta cells, CCK is then secreted to the acinar cells in the exocrine pancreas, which then promotes acinar to ductal metaplasia, leading to pancreatic ductal tumorigenesis. So in order to understand how beta cells aberrantly express CCK, our collaborators isolated pancreatic islets from lean, overweight, and obese mice, and we leveraged several computational tools to identify the cell of origin for CCK-expressing beta cells, characterize transcriptional changes within beta cells, and understand how beta cell adaptation varies across species, ages, and conditions. So first, to identify the cell of origin for CCK-expressing beta cells, we sought to characterize the cell states within beta cells from each condition. And to this end, we used our method AANet, which is a nonlinear archetypal analysis approach that identifies archetypes or extreme states from cellular observations. AANet is an autoencoder that transforms the data into a latent space that's bound by a simplex. This is enforced by optimizing reconstruction and penalizing negative barycentric coordinates coefficients to ensure all data points are defined within a simplex that represents the data. We additionally initialize archetypes with geometrically derived diffusion extrema, which improves robustness in highly noisy and nonlinear settings. So in this data set, given the known diversity of adult beta cell states and how metabolic conditions can modulate these states, we used AANet to map beta cell heterogeneity and identified many shared and distinct programs in archetypes within and across conditions, including one wild-type archetype, one high-fat diet archetype that had high similarity to the CCK-positive archetype in the obese condition. All of these archetypes representing an immature beta cell phenotype represented by low expression of insulin and high expression of dedifferentiation and maturity markers. So in order to understand the dynamics between these archetypes, we next leveraged our approach TrajectoryNet, which is a neural ODE network that interpolates population flows between distant time points. Beyond estimation of pseudotime or branching of populations of cells, we're often interested in inferring trajectories of individual cells for specific cells of interest. So TrajectoryNet learns trajectories by optimizing the vector field of neural ODE to approximate short and biologically plausible paths, approximating unbalanced dynamic optimal transport on the cellular manifold. Considering each data set as time points along an obesity progression, we used TrajectoryNet to trace back CCK-positive cells from obese mice to their cells of origin in lean mice. And we found that wild-type archetype 4, which we described as the immature archetype, was strongly enriched as the cell of origin for CCK-positive cells. This archetype expresses NGN3, or neurog3, which is typically lost in mouse adulthood, but recent works have described adult NGN3 beta cells, positive beta cells, as having regenerative potential, suggesting this beta cell state has the capacity to adapt and proliferate toward the CCK-positive state under stress. So to test this hypothesis, our collaborators performed in vivo genetic lineage tracing, labeling 3 possible progenitors, alpha cells, mature beta cells, and immature beta cells, labeled by neurogenin 3, and performed co-immunofluorescence with CCK to assess overlap and likely cell of origin. Alpha cells showed very little overlap with CCK, and beta cells showed high overlap. And the percentage of overlap for immature beta cells actually increased from 5 to 16 weeks, whereas in the context of obesity, and did not increase in the non-obese condition, arguing that obesity leads to an expansion of the NGN3 positive population, and supporting that trajectory and that prediction that immature beta cells are indeed expanding and adapting toward this pro-tumorigenic beta cell state. We next investigated the genes that were changing along this trajectory, and we saw that genes associated with identity were decreasing in expression, and genes associated with stress were increasing in expression. So to identify putative regulators of CCK among genes increasing in expression, we used a very simple approach by combining known transcription factor target interactions from the TRUST database with genes that were considered Granger causal based on the dynamics in the data. And this predicted Jun as a potential regulator for CCK. Consistent with this hypothesis, JNK inhibition, which is upstream of Jun, in vivo and in vitro showed decreased pancreatic Jun and pancreatic CCK expression in obese mice. In addition, cut and run sequencing revealed that Jun binds to a region 3 kb upstream of the CCK gene, and the intensity of this binding is reduced with JNK inhibition. Data from CCK-expressing tissues and ENCODE suggested that this binding region may be an enhancer marked by acetylation and DNA hypersensitivity, and analyses of human islet epigenomic data shows that this putative enhancer region is conserved in humans and resides at the boundary of a chromatin loop, suggesting higher order chromatin structure may provide an additional level of transcriptional regulation for CCK. Finally, in order to contextualize these stress induction signatures with other physiologic and pharmacologic settings, we used our approach SCMM-GAN, which aligns manifolds of different data sets. SCMM-GAN is a generative adversarial network that is optimized by the GAN min-max objective, as well as a reconstruction loss to enforce cycle consistency and a correspondence loss based on pointwise similarity to enable manifold alignment. In a reanalysis of beta cells from wild-type mice in a different study, cells that were previously labeled as immature by the authors of this study mapped to the immature archetype in our dataset, suggesting shared transcriptional signatures of immaturity. TrajectoryNet then predicted that CCK high cells under an STZ-induced stress trace back to these immature cells, arguing that both obesity and pharmacologic stress induce immature beta cell adaptation to a CCK high state in vivo. We also show that across mice of different developmental ages, postnatal mice are enriched for our immature cell of origin. And in samples from human donors, the CCK high archetype is enriched among type 2 diabetic donors. Altogether, leveraging and integrating these machine learning tools allowed us to demonstrate for the first time that obesity in mice induces a stress response in beta cells, leading to the expansion of an immature beta cell state that upregulates CCK expression by the JNK c-gene signaling pathway via a 3' conserved enhancer. And this axis suggests possible therapeutic targets for intercepting obesity-associated PDAC. And with this, I'd like to thank my collaborators, especially Kathy, Dan, and Mandar, for their biological validation and support with this manuscript, as well as my PI advisor, Smitha Krishnaswamy, and my funding sources, and thank you all for your attention.
So unfortunately, with these super short talks, we don't quite have time for questions. I really encourage you to stick around and ask your questions after the session.